Good morning. I'm Tom Moore, Chancellor of USC Upstate, and it's indeed a pleasure to welcome you to the 10th Annual SC Upstate Research Symposium. This is one of those days where I get to show up and welcome you to USC Upstate and act like I have something to do with this event and this place being the vibrant, wonderful university that it is that can host an event such as this. A lot of people have done a lot of work. Melissa Pilgrim will follow me, and I think she'll probably give some more of that credit. There's credits in the program as well. But a lot of work from a lot of people goes into this event itself, with no mention of the work that goes into the research and the presentations that will happen as a part of this. The connection of USC Upstate to the universities that are represented here and colleges and other institutions here is a wonderful example of higher education across the Upstate cooperating to make the lives of people here and around the world better. I'm going to have to leave before the end of the keynote address for an appointment at my office, but I will stay to hear and see as much of that as I possibly can. I think we're all in for a treat there, and I know you're in for a treat through the rest of the presentations throughout the day. Thank you for being here. It's a pleasure to have you on our campus. I hope you have a wonderful day. Thank you.
Good morning. Can you hear me in the background? Yep, thank you. I don't exactly know where to start with today. As you look around the room, when it comes to an event that is regional in scope, the thanks start where? It takes all of us that are in the room today and that will be here throughout the day to make an event like this work. You hear a lot of buzzwords these days in academia, one of them being building a learning community. And some of what we are showing today is a learning community in action. And so the thanks extend not only to the literal help we had in organizing the event, but also to our presenters, to the faculty mentors, and to all of the participants. Okay, and so it will be hopefully just a day of much interaction and much growth, just in terms of networking, just in terms of how we think or perceive about the world and how we perceive about our connection to not only our home institutions, but our community. Okay. Now, in terms of just practical matters, okay, before I get to the speaker at hand, I have to say thank you to the program committee that helped me organize the event, and I have to say thank you to our sponsors. In your printed program, everybody is listed. Also, we have judges working through the day, and they will be working hard this year. Of the 78 presentations, 50 are student competitors. And so those students, if you're in the room or if you're their faculty mentors, please remember that student awards will be announced today at the Fireside Social. Right now, it's scheduled for outside at the Roel Pavilion, but we may have some rain coming in. So just after lunch, we'll make sure to make an announcement that if we get rained out, we'll be back in the ballroom for the final closing ceremony. We're also doing something new this year, that for those student competitors, if you would like to pick up the judges' feedback on your presentations, during the time frame on the schedule where it says poster breakdown, when folks will be in here after the award ceremony breaking down their posters, there's a judging room, room 319, right back through those doors, where you can actually go and pick up feedback from your judges about your presentations. That way it's also something where a little more is added to the learning experience for you. Okay, so those are some slight changes to years past. In terms of how the day will proceed, you have your long program in your book. Many of the events, okay, are central right here in the ballroom, okay, and so we'll have sessions running after the keynote speaker here and also in room 317, right back behind us. And then we'll also have 2 breakout sessions running in the health education complex, room 2001 and 2039. They're right across the hall from each other, and a nice, beautiful walk if the rain holds off. Okay? And so those are kind of our hubs of activity, so fairly close by for you. And then with lunch right back in here, you'll pick up your box lunches by name in the room right adjacent to us. If you did not have time to register, you're a walk-in, feel free. Our cafeteria will be open downstairs. You'll just have to pay for your lunch as you go in through the line. So there's plenty of food around, plenty of beverage around. The coffee breaks that are listed in your program, there'll be one right here, and there'll also be one over in HEC. So if you know you have to get to some of those breakout sessions in the other building, you'll have refreshments served over there as well. Okay, and so those are kind of the nuts and bolts of the day. And then now I get to a hard part. For those of you who know me well, I tend to find words fairly easily. I can talk a bit. But when it comes to introducing Dr. Guillette, I get a little bit stumped. And I already apologized to him last night at dinner that a word that keeps coming back to my brain, and I'm tired enough that it will come out today, is how strange his academic pedigree is, let alone his life story. Usually it's a pretty straight shot when you go to introduce a speaker. And many of you, when you were reading over his bio, might have said, oh, he's our speaker because look at how prolific he is with his research. Look at all of these publications. Look at all of these books. Look at these titles he has, right? But actually, yes, all that's wonderful. And yes, that means he's appropriate for a research symposium. But you're also thinking research in a very narrow sense, if that's all you capture about the man. Okay, because remember, we represent all disciplines at this symposium, not just the sciences. And at the core of that is creativity of thought, integration of discipline and material. And if you actually look closely at what Dr. Guillette has done with his life, right, he's at the core of an active, engaged, lifelong learner. That is him. He is the embodiment of that. You know, he started, if you think of his bachelor's work, out in New Mexico, and a transition to master's and PhD work in Colorado, working with lizards, to now, if you look at where he's at, he's in the obstetrics and gynecology department over at the Medical University of South Carolina. Can you wrap your mind around that? He's chair of the Hollings Marine Lab's Genomics Center. He's head or the director of the Marine Biomedicine Program. And so he's bringing back a lot of his core research also to how it impacts human health and welfare. So in my mind, it's not just the literal productivity that Dr. Guillette brings to the table. It's his contribution and his representation of lifelong learning. And so to me, I was thrilled that he took the time to be with us today. He has to be at Berkeley tomorrow. And so he's fairly busy. And so I am so appreciative, and he made my year. And so with that, I would like to turn things over to Dr. Guillette. The photographs you were seeing, by the way, those were his own. So he is also a bit of a Renaissance man.
Good morning, all. Can you hear me? I hate being pinned to a podium. I'm a wanderer, so it's going to be a little hard here this morning. And also, as I don't know who gets to talk after me as far as students, but you already have my go for it. You get to give a talk and not see your slides. So I finally got them to convince them to turn it on behind. So for those of you that are looking that way, you can actually look that way. So it is a great pleasure to be here this morning. I have not been to Spartanburg before. In fact, South Carolina, until 4 years ago, was just a kind of spot on I-95 as you drove someplace else. So it's actually great to be here. I had a lot of fun yesterday. I wandered around Spartanburg, took a couple of hours, and spent a day here and visited some of your great sites. And so I do want to thank you. It is an honor to be here, and it's always kind of weird to get an introduction like I just had. And I say that because, and I should say right up front, when I prepare these seminars and I give these talks, I'm not talking to the faculty. Anybody that actually looks almost as old as I do, there's nothing I can do to change you, nothing. But, because I know me. But the fact is that I do hope that the students get some sense of this. And as you heard, I don't think any of us actually plan where we're going to be 25 or 30 years from where we are today. I think even if you ask most of the faculty members here, kind of did they plan to be here? And the answer from most of us is we had some vision of what we wanted to do, teach in a university, teach in a college, do research, but you don't plan to be there. So I'm actually going to give you today part of my history, trying to explain to you how kind of a Colorado mountain boy who grew up literally climbing mountains and doing mountain rescue ended up studying high altitude pregnancy that then turned into studying the evolution of the placenta, which then turned into showing up in the swamps of Florida, because that's where you get a good job, to then all of a sudden discovering something which was quite a bit different. And hopefully that's today's message. So thank you for coming this morning. And my understanding from Melissa last night is she told me that most of the people here were high-end molecular biologists and nano engineers. So about a third of my lecture is math, so it should be okay, right? No, kidding aside. We'll play with pictures. Okay, the students thought they had a day off. You don't. It's a quiz. So, what causes disease? And you have to answer. I won't go on until someone gives me an answer. What causes disease? Come on, if you go to the local Publix or supermarket and people are walking out the door and your teacher says to you, go and interview people and ask them what causes disease. What causes disease? Bacteria. Okay, so you got the first one, germs. Okay, what's the other one? That everyone, what? Viruses, right. Germs, and so you're right. Bacteria and viruses are one. The other one, of course, genes, right? We all know genes. I mean, the fact is that we grow up, anybody took 10th grade general biology, someone said, hemophilia, royal family of England, or in Great Britain, you know, birth defect, etc., genes. But when you go to the doctor and you're worried about your skin, or, quote, they tell you not to smoke, what are they worried about? Basically the environment. But the perception that most people have about genes and germs, it looks something like this. In fact, it's even worse than this. If you actually look at the way federal funding and research goes, the genes should be about 100 times larger, and the Earth should be 100 times smaller than the germs. In other words, the perception really has been, for a very long time, that there's now a whole heck of a lot we can do about our health. Some little things, kind of live healthy, eat healthy, but the reality is you're stuck with the genes mom and dad gave you, okay? And you're stuck with the guy coughing next to you, okay? So one of the two leads to whether you're going to have health or disease. I'm actually here today to argue that the perception's completely different. That the Human Genome Project actually did something fundamental in our understanding of health and disease, and that revolution is taking place, and quite bluntly, most of the medical profession doesn't know it. I mean, it's only been the last 3 years I've been giving lectures to first and second year medical students, and last week I was in Canada giving a talk to the Canadian OBGYN Congress and 1,000 physicians, and quite honestly, 50 of them came up afterwards and said, I'd never even heard what you'd been talking about before. This is amazing. So, that's the message for today. The message today is that the environment plays an absolutely fundamental role in whether, in fact, we are going to actually have disease or health. Now, I spent 25 years at the University of Florida, and if you just kind of remove those images a little bit and kind of shadow it all in, it kind of looks like somebody south of Orlando, okay? So the thing is that this could be a Mickey Mouse hypothesis, but hopefully we'll prove that's not the case today. So let me just show you a couple of patterns that actually strike right to the heart of this. Everyone's heard that breast cancer is going up. I don't have a pointer, so you're just going to have to read the slides, okay? But in the bottom corner, breast cancer. Look over time. Breast cancer is going up. It's going up in every culture, every country that we've looked at, and the fact is that this is going up at a rate faster than population growth, corrected for population growth. So breast cancer is going up at faster than a generational rate. What does that mean? That means that it cannot be inheritance. It's not that we're inheriting this from mom or dad. In fact, what we know today is that breast cancer, only about 12% of the cases are inherited. 12% are because your mom or your grandma or your great-grandma, your aunt, whatever, has in fact breast cancer. And there's some familial, if you will, genetic link. 80% of breast cancers have absolutely nothing to do with familial links of what we think about, okay? Prostate disease, the same. Testicular cancer, the same. In fact, in some countries, testicular cancer now is the #1 disease for young men, which is staggering, okay? And so all of these are increasing at astronomical rates. Take the opposite, the onset of puberty. Now, this is one of the best data sets we have, especially from Europe. It ends up that little girls, and I use that term correctly. I do not believe that a 10 or 9-year-old girl is a young lady yet, okay? The fact is that we now have young girls, 8, 9, 10 years old, going through puberty, actually starting menses and starting breast development. Now, we know that part of that change, okay, is due to diet. We know that part of that is because today we have all the fresh vegetables we want and all kinds of fruit, and so lots of the micronutrients that used to be difficult to accumulate are in fact easily available today. But that's only a tiny part of it. What we also realize is that the environment is part of it, and the argument I'm going to make today is that part of that are chemicals that we are in fact introduced to every day. In fact, we're introduced to them at conception in our lives, function as environmental estrogens and alter estrogen signaling early in development. Now, this phenomenon of early exposure leading to some kind of disease later in life has a new term. It's called the developmental origins or the embryonic origins or the fetal origins of adult disease. And this is a fundamentally different concept, because what happens in this case is we've always known about birth defects. But birth defects, you're assuming that babies come out with missing limbs and not enough eyes and all the rest. In this case, the babies come out looking absolutely normal. You don't know there's a problem until that child goes through puberty or reaches some time in their life later on, when in fact you start to see these abnormalities come online. And in most cases, except for the kind of classic ones, cystic fibrosis, hemophilia, a few others, these are not associated with classic gene mutations, single gene mutations. So this has actually made enough of a splash. It appeared on the cover of Time magazine. And the take-home message for all of this is that a bad start, and in this case, the bad start is the start your mom and dad had, and this is what I'm going to show you here in a minute, actually lasts a lifetime. But it doesn't do it through the classic gene mutation. And in fact, we now know it probably lasts several lifetimes. So if I haven't scared you yet, okay, let's give you a little bit more. So let me explain this little thing to you. And this is really a cool story. This is a detective story. It takes us all the way back to World War II. The winter of 1945, the Nazis actually blocked Holland. Holland actually went through what was called the winter famine. And so it's now called the Dutch famine. And during that 3 or 4 months of winter time, there was no food coming into Holland. And if you were a pregnant mom during that period, then you dealt with it, okay? And the fact is that what we knew now afterwards, and this was actually Robert Bacher actually started examining this data, that the babies born after that famine were all lower weight. They were low-weight babies. Now, we know that's a problem. But what we didn't appreciate is that, in fact, 40 years later, 50 years later, depending upon what trimester mom was in during that famine, depending upon what disease, they actually had a higher preponderance to develop. So if you were in the first trimester, your young, well, actually your children, now middle-aged men and women, were much more likely to develop cardiovascular disease or hypertension or obesity. No, it wasn't because they sat in front of the TV and ate Cheetos. Okay, that helped, okay? But the fact is that there was, in fact, a predisposition to develop obesity, and I can talk about why. If you were actually in the second trimester, you're much more likely to develop pulmonary disease or renal disease, and if you were in the last trimester, diabetes, depression, antisocial personality disorders. Now, you say, well, wait a minute, how does that work? Well, it works because the developing embryo does not develop all of its systems at the same rate. So it ends up that the neurological system doesn't really start to get all connected together until the last trimester, and then, well, my wife doesn't think mine's fully mature yet, okay? She's hoping I'll be a mature 11-year-old someday. I haven't made it there yet. But the take-home is that that neurological development takes until you're in your early 20s. Breast tissue, for example, doesn't reach its full maturity until a woman goes through the first full pregnancy cycle. So there's a whole bunch of developmental issues going on in the body that are not completed when that baby's born. Okay? So that makes sense. We're now starting to say, okay, well, we can understand if you actually have an inappropriate diet or some kind of weird signal at different times during pregnancy, it likely leads to alterations in different kinds of tissues because, in fact, they're developing at a different rate. So let me give you one of the most powerful observations. I work in Africa extensively. I am still sprayed with DDT on a regular basis. Even though we don't use it here in this country, DDT is still used in southern Africa and lots of places to control malaria. Malaria is a major disease that kills lots of babies. We now know DDT is probably killing lots of babies in a different way, causing basically premature cessation of lactation, that is early milk loss, and also spontaneous miscarriage. But here's a really interesting association based on this slide. If you're a little girl, less than 4 years old, and you're exposed to the highest levels of DDT, this is environmentally, that's the blue bar, your chances to develop breast cancer later in life has an odds ratio of 10 or 11. That's 10-fold higher chance of developing breast cancer if you're under 4 when you're exposed. But if you're over 14, your odds are the same as kind of not being exposed at all. So the take-home is that there are developmental exposures that matter. And in this case, DDT happens to be an estrogen, as we'll talk about in a minute. So timing is critical. It's not everything, but it's critical, okay? This happens to be a great white shark that was trying to take us out. I work on these guys. We were in cages doing biopsies, and this one was not happy after we took her biopsy, but that's neither here nor there. So when we think about disease and you think about health, right, we all know that what you eat during pregnancy matters. I mean, every woman in this room knows that, and hopefully most of the guys are not that clueless as well, okay? The fact is that what you eat matters. But what you don't realize is that what's in what you eat also matters. For example, contaminants. We all know that what drugs you take during pregnancy matter. You've all read the little drug thing and it says, don't take it during pregnancy, right? Did you know that personal care products might also, in fact, affect the development of your baby? So these are all things. We knew genetics were matter, but body burden. Did you know that stress might matter? Well, it ends up that no single one of these compounds is important all by itself. It's, in fact, all of these. The developing embryo is an integrated circuit. It brings all of this information together and uses that information in developing. And so it comes out with 2 arms and 2 legs, just like it's supposed to in most of the cases. But whether, in fact, that individual is going to be fertile or subfertile or develop breast cancer or not, or testicular cancer later in life, that actually has to do with what's going on during this phase. So part of the whole deal is something called exposure to contaminants that we now call endocrine disruptors. I'll explain this in a minute. This was actually a hypothesis that was generated in the early 1990s. It was actually started by a woman, Theo Colburn, of the World Wildlife Fund. And she was brilliant enough to basically study the wildlife in the Great Lakes and realized it wasn't cancer that was killing these animals. It was something else. And so she called a bunch of us together. We met in it's called the Wingspread statement because it was actually in the home of Frank Lloyd Wright, designed for the Johnson family on the shores of Lake Michigan. It's called the Wingspread. It's the name of the building. And we sat around, and there were physicians, and there were developmental endocrinologists, reproductive endocrinologists like me, and various other things. We sat around for 3 days, and what we realized is that we were all working on the same problem. We didn't know we were working on the same problem, but we were. And we came up with this statement that basically said, yes, there are a large number of man-made chemicals that have been released into the environment that have the potential to disrupt the endocrine system. Now, what's the endocrine system? Endocrine system is this series of organs that, you know, alligators and fish and we have, okay, that actually make hormones. They make chemical messengers. Now, everybody in this room got the hormone lecture sometime in their early teenagers, okay? And I'm sure everybody here who's a mom and dad gave that lecture, okay, at some point to both their boys and their girls. So hormones are things like insulin, testosterone, estradiol, etc. These are chemicals that come from an organ, go into the blood, circulate around in the body, comes to what we call a target tissue, and there's a receptor. and it makes that cell do something, grow, shrink, you know, make milk, make ovary, make eggs, make sperm, do whatever, okay? That's what the endocrine system is. Endocrine disruption basically occurs at multiple levels. So this is a simple cartoon of the endocrine system. You got the brain, it's a higher level of organization. It goes and makes hormones that make your gonads, ovaries, and testes, or the thyroid that makes thyroid hormone that basically controls metabolism, that makes a series of hormones that then go to your target cells. Endocrine disruption is knocking out any one of those. Not all at the same time, it doesn't even have to be, but any one of those things can happen. And my lab over the last 20 years has documented this at all the levels. Everything from affecting the brain all the way down to literally its effects on the DNA. So what kind of products are we talking about? This is a slide from the National Institutes of Health. And it just, you can see the names underneath, but I don't want, these are the kinds of things we're dealing with. We now, in fact, recognize over 1,000 different compounds. These are compounds that I bet almost every single person in this room was exposed to this morning. You don't have to tell me if you didn't take a shower. If you didn't take a shower, I'm not asking you to raise your hand, okay? If you didn't, then maybe you didn't get exposed. But if you actually were exposed to normal shampoo, personal care products, various kinds of soap, etc., you were exposed to these compounds. And of course, women, believe it or not, are the most exposed in many cases. Why? Because they use more personal care products. Now they're all being told that in order to have your skin look beautiful until you're 99, you should wear sunscreen and everything else, okay, at the same time. Fact is that, yeah, sunscreen is appropriate. I use it. But when I'm going out in the field and I'm going to spend 12 hours or 14 hours in the sun every day, okay? So the take home is that there are multiple chemicals that we now know are endocrine disruptors. So here's the take home at this point. The take home message at this point is kids are different. The fact is kids are not big rats or little adults. They're a developing system. And so we have to actually understand that. The second part is there's a whole lot more you and your health than what you thought. You're not just stuck with what mom and dad gave you, okay, genetically. You can actually do an awful lot about whether, in fact, you're going to be healthy later in life. And that even means for those of us who have a few gray hairs, okay? How we live our lives actually plays a major role. So how about me? Let's tell the story. That was the intro. I got 3 hours, right? Okay, I'm good. You may not know it, but the Medical University of South Carolina actually is a full partner in a marine laboratory. And you go, how in the world does that work? We're supposed to just be no, we're not a hospital. We are a hospital, but we're a research university as well. And one of the things that we actually know is that many drugs, many understanding of what's going on in the world actually come from marine organisms. In fact, many drugs are derived from the ocean, from weird animals, from corals, et cetera, et cetera. And so we're actually at the Hollings Marine Laboratory. The Hollings Marine Laboratory is a joint venture between the Medical University, NOAA, National Oceanographic and Atmospheric, NIST, the National Institutes of Standards and Technology, their analytical chemistry group, the College of Charleston, as well as the state DNR. And so there's about 90 PhD scientists in that building that you see there, along with about another 50 or 60 technicians at various levels that have undergraduate or, in fact, master's degrees. We do high-end research. Most of us are interested in environmental health. If you actually look at the bottom picture, that's my view from my office window. It's a horrible view. Every morning I get to actually, no kidding, the other day I was on a phone conference and all of a sudden I disappeared from the phone conference and they said, Lou, are you still there? And I said, yeah, don't worry, mate, the dolphins are feeding right outside my window. And they said, you're kidding, right? And I said, no, you guys just keep talking. This is much more interesting. But no, and it was a really boring phone conference, but that's neither here nor there. But yeah, so we're basically on James Island. We're on the point, the old Fort Johnson. In fact, they fired the first shots for the Civil War. I'm not a South Carolina resident, so I won't say anything bad about that. But they shot the first cannonballs about 20 feet from my office. But that's where we're set up. And what my lab does is not only human research, but we do something we call sentinel species work. Now, the concept of a sentinel comes out of the Middle Ages, right, or even earlier than that. It's the guy standing on the wall trying to make sure that there's no fire in the village or no one's coming to raid. It's the watch and warn system, okay? It's that, and we now use wildlife for a number of reasons, because they are, in fact, our watch and warn system for the environment. They're the ones that having to live in the environment every day. They're eating in the environment every day. Their cheese is not coming in from France, okay? Their wine's not coming in from California. The fact is that they're eating what's there. Many of them don't get up and fly away. They live there their whole lives. And so, for example, in Charleston Harbor, we do the dolphins. We also do the sea turtles down off of Merritt Island. You can actually see the bottom picture. That's Cape Kennedy, Canaveral, right behind my buddy there, Russ Lowers. We do alligators there. We do great white sharks off the coast of South Africa. And today's story is about alligators. We've been using them for a long time. Why? Because we can actually intimately study this interaction between environment and genes. Now, in order to use a sentinel species, you actually have to be able to interpret that data. And as many of you actually know, we used to take little canaries down in coal mines, okay, for 100 years to do the quality of the air, right? And the canary was small enough, even though its lung was a bit different, methane killed it. And so these two miners are looking at it and say, yeah, this dead canary means something, but we're not sure what it is, right? Unfortunately, that's pretty much us, okay? I'll tell you bluntly, that's pretty much us, whether it be climate change or whether it in fact be this topic. We actually see what's going on around us, but we really haven't been interpreting the data. And so again, hopefully today it will help you start to understand this. So, Colorado Mountain Boy, 1985. I go to the University of Florida. I'm there about 6 months. I've been working on these high altitude lizards on the top of the mountains, Mexico, in the Andes. Great place, love going there. All of a sudden, knock on the door, guy in a uniform. Ooh, what did I do? He goes, we hear you're a reptile expert. Not. Reproductive biology, maybe. I work on little lizards. They said, well, we want you to work out the reproductive biology of the American alligator. I said, you're kidding, right? I said, there's 100,000 people trying to work out the reproductive biology of humans today, and we're going to work out the reproductive biology of how much money do you have? Oh, $20,000. How long? 3 years. Not going to happen, okay? More importantly, it's not going to happen because if I get bit by my little lizard, I get a blood blister, okay? If I get bit by these guys, they say, no, we're going to set up two teams, the outies and the innies, okay? You're going to lead the innie team, the skin in, and these are the wildlife guys, we'll lead the skin out. Sounds great. So we went out on warm summer nights, and we started counting alligator eyes. So all those dots are alligator eyes, okay? And what's amazing is you can actually not only say, okay, how many animals are there, but you can actually get some idea of recruitment, that is reproduction in the population. How many babies do you have? How many teenagers? How many adults do you have? Then you can fly in a helicopter over, how many nests do you have? And you can start doing some fairly sophisticated ecological models and start to put together an idea of what's going on in this population. Now, what's really important also is that you have to be at a university with undergrads, because they're the ones you put on the front of the airboat, okay? So as you're running across the lake, they have to make a snap decision, okay? If it's 5 feet or under, you grab it with your hands. If it's over 5 feet, you noose it. The joke in the lab is that if you make a mistake, your dissertation is going to take twice as long to type, okay? So I will report, I know the provost doesn't want to hear this, but I will report that a number of my students did go to the infirmary with animal bites and duct tape bandages, but it's hilarious. So the nurse says to one of my students, that's not a dog bite. He says, I didn't say it was a dog bite. I said it was an animal bite. He says, well, what animal? And I said, alligator. And she says, were you molesting it? And he said, yes. And so we got very well known in the infirmary for molesting alligators. But the take home is that we would go out, we'd grab these animals, we'd actually take blood samples. We had to develop all these techniques of how do you get a blood sample. So it's a vet check. It's what happens when you actually take your dog or cat to the vet. How do you get a blood sample? How do you get a urine sample? How do you get a tissue biopsy? How do you assess their health? And then the goal is, how do you make sure that you let them go, okay, and they're as healthy, other than being a little stressed, as when you caught them, and you're as healthy, okay, when you release them. So same stuff Melissa does with her snakes and all of us. I mean, the point being here is that we want to release these animals unharmed, but also be able to get information to say something about the quality of the environment. I never did lose an undergrad, okay, ever. But what we did start to realize is that these populations were not growing at the rate that was expected. This is 1986, 1987. This was when the animals were completely protected. There was no hunting going on, little poaching, but we didn't worry about that in the middle of the night. Those guys had guns, we didn't. So the take home is, we had to start going, if there's a problem with recruitment, that is numbers of young coming into the environment, then we actually have to go to the site of recruitment. Site of recruitment is reproduction, and that's a nest. So what you do is you get the National Institutes of Health to rent you a helicopter, and you fly around on a summer morning, because you can see one of my students standing on my airboat seat there. The seat's 5 feet off the water. He's 6 feet tall, and you can see the grass is like 11 feet tall. Now, somebody in this room actually knows this environment really well, because this is Lake Woodruff National Wildlife Refuge, which is right outside, well, same place they did a whole bunch of snake work. But if you actually go back into that marsh, if you look at it from the air, that's an alligator nest. About 6 feet in diameter, about 3 or 4 feet high, guard pool around it. See that little trail coming out on the bottom? That's where you send the undergrads in, okay? Because they're a renewable resource at a university, right? So you send them in. You do let them use a Jesus board. Do you know, so they can walk on water? I mean, this is actually just a 1x10 that you put on the grass. You still are in knee-deep water wading in there. So it gets a little exciting. There's one final story I think I have to. So I have this young woman who's been I'm an equal opportunity employer. You want to do it? I'm glad to teach you. Doesn't matter. Sex had nothing to do with whether you could do this job or not, okay? So she said, oh, she'd been working with me for 2 years, 5'1", 120 pounds, but she's going to do it. And I said, let's do it. So she'd been in my lab for 3 years. She climbs out of the airboat, slapped the board down, kissed the board, and she has the pole, because you see next to the nest, Mama's there, and Mama defends the nest. So the first person there is supposed to do one of two things. One, determine whether Mom's there. If Mom is there, noose her. In other words, catch her so we can control her. So she gets on the first board. I hand her the second board. She slaps it down. I stand onto the first board. I'm carrying all the egg kit. That's that thing you can see in the bottom, right? And all of a sudden, the next board's there. Mama comes up next to the board. And she goes, whoa! And she takes the pole and knocks me down, steps on my back, and then jumps back into the boat. So I'm laying out on the water looking at this alligator. She disappears. I climb back in the boat. And I said, what did you just do? So, oh, no. I said, you stood right in the middle of my back. Oh, no, no. Look at the footprint. And so, sure enough, that T-shirt was her graduation present. I had it framed. But she was a superb student. She actually then went back out and did what she was supposed to do. But that's a nest. We go into the nest. If we catch Mom, we try and catch her. There's the nest. You actually get the eggs. We collect all the eggs, because if we don't, the pigs, the wild pigs and the raccoon come in and get all the eggs right after us, because they follow our scent in. We bring the eggs back to the lab. We incubate them under controlled conditions. Give you a little statistics. About 35 to 50 eggs in a nest. Mamas are about 6.5 feet when they reach sexual maturity. That takes about 12 years. We now have confirmed data from marked animals that these females will lay eggs for up to 34 years. So we're talking about a lifetime that's very similar to ours. Sexual maturation around 12 to 15 years of age, potential reproductive lifetime of 30, 35, probably even 40 years. Total lifetime easily 60 years or more, okay? So that gives you some idea of long-lived species, sensing the environment. Most of these females we now know, after 35 years, they're 100 feet from where we caught them the first time. So their territories are really quite small. So they really are sensing the environment there. It's not 100 miles away and then they come back, okay? So the take home is that we collected all these eggs. To date, we've done almost 500,000 eggs. We take 99% of those that hatch and take them back. Mom takes them back. We take a small sample to actually do the research. But what we found was that even on our best lakes in Florida, at that point, we were only getting 50% hatch rates. Now, on some of the lakes, we're up to around 80 or 85% hatch rates. Our worst lakes were giving us 10 and 15% hatch rates. Now, under controlled conditions, that's not normal, okay? I've worked on Nile crocs in the Okavango Delta of Africa, which was one of the more pristine areas, 95% hatch rates. Northern Australia on salties and freshwater crocs, 90% hatch rates. That's really where we should be. And we have a couple of lakes now in Florida that does that. We have no place in South Carolina yet that we found. We're now working in the Ace Basin. Hopefully we'll actually, looks a relatively pristine environment. So here's the logic. Logic. You have 35 to 50 eggs. Let's say every single one of them hatches. We know that 90% of those offspring will be dead in the first year, okay? Picked off by osprey, eagles, great blue herons, et cetera. I mean, really is a very steep survivorship curve. So here's my logic. Who cares about what kills eggs? What I want to know, if a female reproduces for 30 years, for zero population growth, she only has to produce how many offspring? Right? She has to replace herself and her partner, just like we, right? So it's 2 kids. She has to have 2 that reach sexual maturity. That's all she needs. And they need to be healthy. Okay? So instead of actually feeding them little baby alligators, we feed them eggs. Okay? So my question became not what killed the eggs. I mean, there was an issue there, and I'm not belittling it. But what I wanted to know is when this little one came out, 12 years or 15 years or 20 years from now, would she or he be a healthy representative of that population? The problem is no one gives you 15-year research grants. Okay? Doesn't work that way. So we had to develop a whole series of techniques, and in part of that was the question that I also had is, could we predict similar kind of lifetime problems for our own species? Do we have to wait 15 years to figure out whether, in fact, a little girl or a little boy is going to have a fertility problem, et cetera, et cetera? So that's the logic. So what we in fact did was to start to study a whole series of lakes in Florida. I'm primarily going to talk about just 2, Lake Apopka and Lake Woodruff. They're down in central Florida. I'm not going to talk about Kennedy Space Center today. There's a whole nother series of studies. And as I said, we're also working in Yawkey in the Ace Basin here in South Carolina. Started that work 4 years ago when I actually moved here to South Carolina. Lake Woodruff is a national wildlife refuge. Never had modern agriculture around it, never had a modern municipality putting stormwater, sewage, or anything into it. In contrast, Lake Apopka is considered the more contaminated lake in Florida. Lots of agricultural chemicals, lots of stormwater runoff. But let me put this relative to you. So about 6 months ago, I'm in Sweden at the Karolinska Institute. I'm giving this seminar. And all of a sudden, there's guys in the back of the room, and they're going at it, and I thought, man, I've just insulted somebody. Something's really going on. And finally said, okay, stop. Okay, what's the problem? They said, just ignore us. We're like freaking out back here. I said, well, why? They said, well, you're Lake Apopka. You're giving us all this stuff. You're, saying it's highly polluted, etc. We're trying to restore the Baltic to your Lake Apopka, okay? The levels that we're trying to get to is there. So the fact is that it's relative, right? I mean, there is contamination, but it's not a cesspool. That's important to know. So here's 20 years of data on one slide, okay? And here's the take-home. The take-home is if you hatch out, that is, we have hatch problems, but the little top panel, if you hatch out, we can already see the day you're born that you have abnormalities of your testis or ovary that we can actually see with our eye pathologically. We already know that you're not making hormones at the right level, and that's related to you already have gene expression that's different. And you don't grow out of this. This is not baby fat, that somehow by the time you reach a teenager or you reach adulthood, that's all gone. So it persists. That's all that slide says. And now let me show you a little bit of data. So the question is, what was the association? Well, I'm not a toxicologist. Okay? Even though I'm going to talk about contaminants today, that's not my training. My training, I'm a developmental endocrinologist. I'm a developmental geneticist. My question is, how do you in fact get health and disease? And so when we went in there, we thought, well, maybe it's temperature, maybe it's what they eat, maybe it's weird genetics from mom and dad. Maybe it's a founder effect. You had some weird individuals that showed up at this lake because they were hunted and they were almost extinct in some of these lakes. On and on and on. Nope. We did basically 10 years of work. So we started saying, well, wait a minute, what is in what they eat? And sure enough, when we started looking at that, we started to find some really intriguing things. Now, this is a very complicated slide. You can ignore almost all of it, other than the take-home is the following. There are heavy metals that are present in everybody. Okay, these are from industrial activities. There are these contaminants, and what you can see is I made a stacked bar graph. It says Apopka, and there's that big bar with a great giant pink block on it. I put it as a stacked bar graph because we're not exposed to one chemical. Those are all the different chemicals that we can measure in that individual, okay? Now, I just want to give you a tiny little sampling. DDT we've already talked briefly about, but in 1952, DDT was shown to be an estrogen, okay? But it was assumed by my field, endocrinology, that it was really unique and rare because we taught endocrinology as locks and keys. We basically said the hormone, okay, is this chemical and the receptor is this lock. So it's the key and the lock. And you only get certain locks and certain keys to come together. I'm here to tell you that's baloney, okay? It's not the way the system works. The way the system works and the way the system evolved was that if you were talking about endogenous, that is normal chemicals that should be in the body, the system is designed to be able to recognize only estrogens, okay, and not recognize androgens like testosterone or thyroid hormone or insulin. In contrast, insulin receptors recognize insulin. They don't recognize estrogens and androgens, etc. But nothing actually told these systems that they weren't supposed to recognize all this other crap that we've actually released into the environment, okay? So DDT is a perfectly good estrogen. The major metabolite, of DDT, is something called DDD. That's the next one down. It ends up, it's been sold as a drug in this country for 50 years, called mitotane. It was used to treat adrenal cancer for 30 years before we got better drugs. This is a metabolite of DDT, okay? It's still used in the vet world to treat Cushing's disease because it blocks the adrenal steroidogenesis, your adrenal glands, and it blocks those hormones, okay? It's a drug. It's a metabolite. In fact, most of us have this detectable in our bodies. Finally, DDE, which is the major metabolite of DDT, is an anti-androgen, blocks testosterone. In fact, it's as potent as some of the drugs we use to treat prostate cancer, okay? So there's not a hypothesis that there's endocrine disruptors. They're real. They're out there. And they're ubiquitous. There's a large number of them. The question is, do we have enough in us to actually make an effect? And that's really what the central question is. So, as the young folks here, just because you find a problem and you find a contaminant doesn't mean one causes the other. Just because you stood on the corner, okay, and an accident took place didn't mean you caused the accident, okay? Now, you might have if you jumped out into traffic and jumped back, but we got to prove that. So in this case, we have to actually try and put this together. So rapidly through here, I'm going to take one example today. It's a developing embryo. I'm going to look at one organ, the ovary. I'm going to pick on women today. I'm in a department of OBGYN. I could have done the penis study today, but we didn't do it. This is South Carolina. I have to be careful what I talk about. No, I'm kidding. I'm kidding. So here's a biopsy of the ovary. And the top panel, that's a normal ovary. You see that big round thing in the middle? That's an egg. Okay, that's an oocyte. In the middle of it is a nucleus, okay, and it has a little half moon, dark little purple half moon. That's called the Balbiani body. That's messenger RNA, proteins, etc. That's normal. One egg, that is one oocyte per ovarian follicle. That's what it's supposed to be. Down on the lower panel, you actually see what happens if you're exposed to environmental contaminants. That is abnormal. Instead of having one oocyte, you have multiple oocytes, strange looking. Some of them have two nuclei. And there's a pathology that's called the multiocytic or polyovular follicle. Now, we don't have that pathology because Lou Guillette found this in alligators. We have it because it was found in little girls and in mice that were exposed to a pharmaceutical estrogen called DES, diethylstilbestrol. And in fact, in the 50s, 60s, and early 70s, we treated in this country 7 million pregnancies with DES to try and prevent premature birth. It was the absolutely wrong thing to use. It had no effect. Even the data wasn't there to support it, but it was just one of these things that the medical profession at that time thought they could improve on pregnancy. They didn't think pregnancy was good enough at that point. We're still not learning that lesson sometimes, but that's neither here nor there. The take home is that every little girl that was exposed in utero to DES has an abnormality of her reproductive system. Many of them have subfertility or infertility. Many of them have this system called the multiocytic follicle or the polyovular follicle. These eggs can be ovulated, they can be fertilized, but in almost every case, the embryo dies in what we call early gastrulation, about 100 cells to 150 cells. So in most cases, you have fetal or embryonic loss, okay? Or you have infertility, that you can't even fertilize the egg. So our hypothesis is, in fact, that the problems that we're actually seeing in our alligators are not due to toxicity. That is, that mortality that we're seeing is not because their toxic chemicals are high there. It's because, in fact, we've messed up the egg to start with. So if that's the case and it's estrogen-based, how do you get an ovary? So I'm here to tell you one thing, undergrads. We have not found all the important stuff. I know you go into classrooms and professors tell you everything is known. Bullshit, okay? The fact is that there's Nobel Prize winning experiments needing to be done today. We are in 2014. I still cannot tell you, okay, the genetic pathway to a normal ovary. I can tell you 5 or 6 genes that are critical for the ovary. I cannot tell you how you get a normal ovary. So how can we understand an abnormal ovary if we can't even figure out how you get a normal one? Okay? That's just one. You want it? I'll give you another 50 different questions. Not a problem, okay? And I'm sure your profs can do the same. The point is that there's really lots of really important things. So, question is, how do you in fact get an ovary in an alligator? Well, what's cool in an alligator is you get it by temperature. If you incubate an egg at 30 degrees during a 72-hour window, you get 100% females. So temperature drives sex determination. But it ends up, and that's different, if you do 33, you get males, but you can sex reverse them by giving them estrogen. Incubate at a male temperature, give them an estrogen, make ovaries, okay? Really cool, okay? Can't do that in human beings, okay? But, oh, if only, okay? The world would be all females. I know, they just keep a few of us around for entertainment, but that's neither here nor there. Okay, the take home is that although temperature drives that, the genes that are involved in the formation of the testis and ovary are the same genes as are in us. So the switch in us is a gene called SRY. It's not temperature, but downstream the genes are the same. So all the genes are the same. And one of the critical ones in the ovary is something called CYP19, the very bottom of that panel. CYP19 also has another name called aromatase. So it ends up that we have the same genes. But why would NIH fund alligator work? I mean, are you kidding me? Why don't we just do humans? Well, because we don't do experiments on humans, okay? That's unethical. And the fact is that it's really tough to do human research on this topic. Why? Because when a baby alligator is born, she has no ovarian follicles, okay? Her ovary is still undergoing all of the cell division needed to make all the eggs. In contrast, when she's born, she's already losing ovarian follicles. In fact, at 6 months in utero, a little baby girl has more ovarian follicles than she'll have the rest of her life. From that moment down, ladies, it's downhill, okay, as far as ovarian number is concerned, okay? So the take-home is that if you want to study this process of abnormal follicular development, you can't do it in utero. And in a mouse, the final processes take place in the last 2 days, 3 days after birth. So all of those early processes are all done. That's why they fund me to do alligators, because I can make all females all the time. I can make all sisters. And, in fact, the process is still ongoing. It takes 3 months after the baby's born. But on top of that, it's the same genes as I've shown you, and one of the key genes is this thing called P450 aromatase, or CYP19. Now, what it does is converts testosterone to estradiol. I know you've all been actually said or told that there's these female hormones and male hormones. No such thing, okay? Every man in this room makes testosterone and makes small amounts of a hormone called estradiol 17 beta. If you learn nothing else from me today, there is no hormone called estrogen, okay? All the women's magazines are wrong, okay? It's a class of hormones. The principal estrogen is something called estradiol 17 beta, much more sophisticated. Sounds like you actually know what you're talking about when you use that term, okay? So in contrast, okay, women have very high levels of aromatase. So therefore, they take most of the testosterone they make. They don't become testosterone poisoned like guys do, okay? And they convert that to estrogen. That's what makes them smart, okay? So that, and guys is the same. I mean, we're only bright because our testosterone is converted to estrogens in our brains, okay? But we don't do it very effectively, obviously. But the take home is, okay, P450 aromatase is in fact a key. And what we've actually learned by knocking out the estrogen receptor is that this is absolutely essential. That is, estrogens are essential for not only the mammalian ovary, but it's also essential for the human ovary and the alligator ovary. So how do this all work? Well, this hormone has to come into the cell and has to bind to a receptor called the estrogen receptor. And this receptor is really cool because it inserts itself directly on the DNA. It's a transcription factor, and it turns on specific genes. 2,000, by the way. In 2 hours, a single injection of estradiol will turn on 2,000 genes in a cell, okay? And it does it, comes into the cell, binds, turns on the genes, and what's really cool about that receptor is that it's found in the exact same chromosome location as us. It's on the short and the long arm of the chromosome number 1. Its structure is the same. It has a DNA binding region. That's that little green bar, this little cartoon there. See that 100? That means that the amino acid sequence of the alligator estrogen receptor and the human estrogen receptor are 100% identical, okay? Not 99, 100. So we're talking about 200 million years of evolution, doesn't matter, okay? The fact is that estrogen is absolutely essential for you to be sitting here, okay? And so the DNA binding region is highly conserved. The hormone binding region is highly conserved. And so what we can do is we can take that receptor, we can make a plasmid, we can insert it in a cell and make a molecular machine. And now we can take any receptor from any species, put it in a cell, and we can use it as a test system. We can take a test chemical or a hormone, put it on the cell, put a little response element in there, and in fact, if the hormone binds to the receptor and turns on the gene, it glows, and you can measure that. And if you have more and more hormone, you get more and more glow, and that's what that increasing bar graph shows you. So here we are. It looks like it smells like it tastes like it. The fact is, this is an estrogen receptor. It's just like the human. In fact, it works at the same level. And if you actually know math, that's 10 to the minus 13, that's 10 to the minus 12. We're talking about picomolar concentrations. We're talking about incredibly low concentrations that this hormone works. And then all the same genes are involved in the process. So not only do we have an estrogen receptor, but then we say, okay, if we look at the developing ovary, or do the right genes come on? And all this panel shows is called a heat map, for those of you interested. The upper panel, you have the things that are red. Those are the genes that are on. The blue, those are the genes that are off. So you can see the female, there's a bunch of genes that are on. Those are the ones that are supposed to be on, okay? In contrast, you can see they're off in the testis. That's the way it's supposed to be. So we actually know same genes, downstream of estrogen, all the system basically is telling us the alligator is very similar. So now, the next point. Can we, in fact, do something to ask whether this ovary will respond appropriately? So here's a cool gee whiz wow. It ends up that puberty has nothing to do with your ovaries and your testes. Puberty actually happens to do with your brain. It's a central nervous system phenomenon. So if you give a baby girl or baby boy some kind of exogenous or some kind of steroid, in other words, for some reason, let's say a baby girl is exposed to an estrogenic drug, her uterus will in fact grow, her breasts will grow at 2 and 3 years old. And that's happened in a few places with environmental contamination. Okay? The fact is that the system, that is the ovary, the uterus, the breast tissue, it's all ready to go. Okay? It's just waiting for the signal. The signal has to come from the brain. The brain doesn't respond until you're 12 or 13 or 14 years old, or in some cases 7 or 8 or 9. Okay? So what we do is we kind of play that game. The hormone that comes from the brain is something called follicle stimulating hormone, FSH. We can actually inject that, and we can ask, okay, just short term, single injection, does in fact the gonad respond? Now this is the exact same thing that happens when a young woman, relatively young, woman or man comes to an infertility clinic. One of the first questions the physician asks is, if I in fact challenge you with a gonadotropin, this is what it's called, or FSH, will your ovary or testis respond like it's supposed to? Can I see the increase in hormones? Because if not, then I actually now start to get an idea of where the problem is at the ovary versus the brain. So you can start teasing out, how do we treat this person for infertility? So we did that. And this panel, all you have to do is worry about the top and the bottom, but you just ignore the panel. I'll just tell you. The thing is, on the top panel in red, what it basically says, you give the injection of estradiol, these little females all make estrogens. They make estradiol, and it goes up over time. That's what's supposed to be, okay? See the panel in blue? See that white bar? Basically says it looks like it's increasing. That's if you're exposed to contaminants. That hormone's testosterone. That is not supposed to happen, okay? Ovaries are not supposed to respond to FSH by making testosterone. That's a pathology. If you look in the bottom panels, especially the bottom, for me, right, okay? That's aromatase. That's CYP19 again. It ends up that white panel, you see it doesn't change. That's why you, in fact, have testosterone at the top, okay? So, in fact, aromatase was supposed to come on. That's the black bars. That's what happens in a normal female. And that's why, in fact, they make mostly estradiol and they don't make testosterone. Does that all make sense? okay? Well, guess what? That's identical to a condition in humans called polycystic ovary syndrome. And PCOS is actually epidemic in this country and in Western Europe. And it's increasing. What's interesting is, as you see the little thing in the middle with the question mark I put on it, up until, in fact, a couple of years ago, since we started doing our work and started finding all this stuff in alligators, the assumption was that this was due to a genetic mutation. And about 8% of the cases there are identified with a genetic mutation. The fact is that 80% are not. And we now have evidence that, in fact, this is probably developmental in origin, that it actually happens early in life, and that it's likely environmental. And so how does that work? Well, here's a really cool thing, and I'm not going to go into details. Some of you, I put this up for the biologists here, and I apologize to the others. But how do genes work? Well, genes are actually a lot like words, okay? Think of it as actually having a dictionary. We don't have 300,000 genes. We have 25,000. So the fact is there's not going to be a gene for Alzheimer's, okay? All of that crap that's in the newspapers are wrong, okay? What we're in fact going to find is multi-gene problems. In fact, most of the diseases we're dealing with today are multi-gene phenomenon. And so what's going to happen is it's how genes are turned on and shut off, how loud they are on and off that matters. And the way you actually regulate that is one of the ways is that the promoter, just in front of the gene, there's a little region called a promoter. It's whether you in fact put stickies there or not. And what I mean by that is that if you take little methyl groups and you stick them on the promoter, it's kind of like taking rubber cement and putting it in your dictionary between the pages. It sticks those pages together and they're not available to you, okay? Now the reason that the genome does that normally is during embryonic development, if you're going to be a muscle cell, you don't need the genes that make you a nerve cell. And so why do you have to read the whole dictionary only to find the pages that you need for muscle cells, okay? So the take-home is that you have to have a mechanism that actually reduces your gene from 25,000 to some reasonable series of units that can be used, okay? Now, I'm being a little simplistic, but that's neither here nor there. We can go into more details as people need. But the point is that what we've actually found is that to be a boy, to have a testis, your aromatase promoter is what we call hypermethylated. You stick a bunch of methyl groups on there because you don't need aromatase. You don't need a lot of estrogen in your ovary. Sorry, you don't have one. You don't need a lot of ovary in your testis, okay? You need aromatase in your brain, okay? That's controlled differently. So what you in fact do is hypermethylate. That's what these blue bars are showing. There are 6 different regions in the promoter, and we published this just last year. To be a male, you hyperpromote those or hypermethylate those. To be a girl, the ovary, you hypomethylate them, okay? There's lower methyl groups. That's pretty cool. That's how the system works. In fact, we don't even really know this in humans yet. We have data in my lab now suggesting this is the way it works, but we haven't published it yet. So what happens if you're exposed to contaminants? Well, it ends up that your ovary and your aromatase promoter gets hypermethylated. You're halfway between a girl and a boy, okay, as far as that gene is concerned. So when the FSH comes down, says turn on, you're kind of semi-stuck together. You don't respond as well as you should. And so you don't make the gene, therefore the gene is not available to be transcribed. You don't actually have the protein. You don't have the enzyme to make the hormone, okay? So where are we? We actually are further in the alligator than we are in humans. And so what we're doing right now is that we actually have a study, in fact today, my students, some of them were actually at a fertility clinic. We do almost every day. And as we harvest eggs, the physicians do, for doing in vitro fertilization, the throwaway are the cells that are all around that egg called the granulosa cells. And we harvest them, and we're doing what are called transcriptome studies. We're measuring all of that. We're doing the methylome. We're looking at the methylation of all those genes. And we're also doing what's called the exposome. We take the follicular fluid, and we measure it for all the kinds of contaminants and metals and personal care product chemicals and all of that. It's a huge study. We're just 1 year in, but we're trying to get some idea of what's going on here with the human so that we can actually match it back. But why are we looking? Because the alligator has told us that this is what we should be looking for, okay? So this is not the only disease. And as I mentioned, polycystic ovary syndrome is there at the top. This was actually from a paper that I actually brought together the Johnson Family Foundation, gave me money to do this. We read a couple of thousand papers, and what we were shown is that all of those diseases are diseases that young women come to their gynecologist with, or their obstetrician, and they have complaints. And up until recently, what we've actually said is, well, what's your job? We don't ask them, well, what was your mom's pregnancy like? Where was your mom living? Where was your grandma living? Okay? Because we're now starting to realize that that history is probably as important as the history of the young woman who is basically sitting there in the clinic at the time. So that's the take home. So, final. We all start off as indeterminate beings. We actually have a gonad that can be either an ovary or a testis. Depending upon the signals that come in, our cells in that developing gonad will become either an ovary or a testis. What's interesting is that cells that make the hormones, called the thecal cells or the Leydig cells, are exactly the same stem cell. It depends upon the signal they get of whether they become a thecal cell in an ovary or whether they become a Leydig cell in a testis. So my hypothesis has been, and I think we're starting to get evidence for this, what happens if you're exposed to an environmental chemical that actually gives you the wrong signal? Could it be that your thecal cells in the ovary actually think they're Leydig cells? So they're actually making testosterone when they're not supposed to be. And that's what we're in the middle of doing right now. We're trying to figure that out. Because we like to see sex as a digital world, zeros and ones. You know, you're either male or female. And you are. But the reality is, at the cellular and genetic level, it's a lot noisier than that. And we're suspecting that's the case. The other take home is that we've spent most of our lives in the world of genetics thinking about genetic determinism. That is, we're thinking about disease based upon mutation. What we now know is that that probably only explains 10% or 12% of the diseases we're dealing with. The other 80%, okay, are likely associated with something we're now terming epigenetic determinism. That is the environment, and that could be contaminants, it could be nutrition, it could be stress. There are multiple factors actually alter how our genes are turned on and shut off. So really, when you think about health and disease and our health and disease is actually a combination of both what we inherited from mom or dad, genetics, as well as the epigenome that our parents basically established during their lives. And we ourselves, our health today, is a combination of both our genetics as well as our epigenome. And that's part of our lifestyle, how we're living our lives, what we're exposed to. And the final component of that is that our kids will in fact be the product of our genome as well as our epigenome. Now, how do we get to all of this? By doing basic science. I'm talking to the choir here, but this is the kind of birthday card you get when you do my kind of work, right? So Daisy, Bubbles, and Elrod suspect someone slipping super hormones into his feet. How do I know this cartoon was drawn by a guy? He has no idea what the udder is for, okay? You got a second set of mammary glands there. That's impossible. And never took general biology from me because you got a bird with mammary glands, okay? So why do I put this up? Well, I can actually take an estrogen and I can redirect the sex of a developing alligator to make it female. I can't do that with humans. But if I take a baby female alligator and a baby female human and I give them an estrogen exposure at the wrong time, then I get the same pathology of the ovary. So again, what is normal? Understanding normal allows you to understand abnormal. And many times you can't, we're in a world today where everyone wants focused research. Go out and solve this problem. Okay, we can do some of that, but an awful lot of the discoveries that are being made in science today, in engineering, are just because people are going out and asking fundamental, creative questions about the world around them. And that, in fact, is how we get forward, okay? And what's even more important, what you've seen hopefully today is chemistry, biochemistry, genetics, developmental biology, ecology, basically wildlife management, conservation, medical science. Those are all my partners. I can't wear that hat, okay? That's not one hat. So modern science is interdisciplinary, it's cross-disciplinary, it's teams. And as a final component of that, it's communication. If you can't talk to your colleagues and explain what's important, then they won't understand it. Now, I realize that for some of you, you're freaked out. You're going to go home and throw everything out, move to a log cabin in the woods. Doesn't work, okay? It's contaminated too, okay? So here's just a couple of quick things. The first thing is moderation. And what I mean by that is that if you're going to use chemicals in your life, spend some time becoming educated, okay? Some of us actually are alive today because of the drugs we have to take. That's okay, but every time we take that drug, we release it into the, into our urine or our feces. It goes into the sewage treatment facility. It comes out the other end. I don't know if you know, there's an incredible story discovered in England. Women take birth control pills. Birth control pills go to their liver. They have a little sugar group put on it called a glucuronate. It goes in, inactivates it, goes in the urine, goes to the sewage treatment facility, goes to the bacterial beds. The bacteria could care less about the estradiol. What do they want? The sugar. They chop the sugar off. What comes out the other end of the sewage treatment facility? Activated estrogen. So all the male fish below the sewage treatment facility have yolk protein. They literally have ovotestes because they were exposed to estrogens. Pretty cool little story. Neither here nor there. Okay, so take home is that if you're going to use chemicals in your house, etc., think about it. You've all heard about BPA, plastic. If you haven't, your head's been stuck someplace, okay? The fact is BPA. Do you know that BPA was actually designed as one of the first pharmaceutical estrogens by Sir Charles Dodd, who won the Nobel Prize for doing that? It ends up that it wasn't strong enough, so he metaphorically put it on the shelf. 10 years later, polymer chemists actually needed something to link their chemicals together. BPA looked great. 40 years later, we're shocked that estrogens are leaking out of plastic. Next week, I'm in England talking to polymer chemists, okay, to try and get them to realize they got to think a little bit more before they do their work. The take-home is that think about the chemicals you're using. And I know this is naive and it sounds very 1960s. I look like I come from the 60s. I do, okay? But the fact is that you really should treat the Earth like a loved one. This is Earth Week, right? We had Earth Day at MUSC just the other day. The fact is it's the only one we got at the moment, so it'd be worthwhile for us to actually think about how we're using it. And finally, of course, the way to do that is to try and support companies and businesses that actually have the same ethic you do, that are thinking about the world and about your kids. So I thank you all. As you realize, I represent a whole bunch of people, and you can see my people. I've had over 100 undergrads in my lab over the last decade. These are grad students and postdocs and several undergrads. The guy up in the upper corner with the uniform on, that actually was Woody Woodward. He's the one that got me in the hot water way back when. He just retired last week. The guy in the middle is me. But the two guys, the older gentleman is Howard Byrne, who is my academic mentor, National Academy member, died a couple of years ago. Howard was the guy who found the DES daughter. He discovered it in a mouse 10 years before humans and was screaming and yelling, and they said, oh no, humans are different, humans are different. Well, of course, 10 years later, they weren't different. So what I do in my lab is to try and figure out how she makes a healthy one of those. I think that if the environment is not healthy for her offspring, it's unlikely it's healthy for our offspring. So I thank you all for coming today. I actually have to disappear right around lunchtime today. I apologize. I do have to be in Berkeley tomorrow to give a talk at 10 o'clock in the morning. But I will be around a little while, and so certainly the students, anybody would like to talk to me, I'd be glad to talk to you. So thank you again for inviting me.